I'm really honoured and excited today at this session of the International IVF Initiative. For those that have been joining to these for quite a while, you will know that there are previous sessions and videos available, and you can go to ivfmeeting.com and see previous sessions. There's some amazing series of lectures that have been held, and I hope today stands up to the ones that we've done in the past. Thanks for logging in. It's great to see everyone joining in. During the session, so I encourage you to ask questions using the Q&A section of the conference service and not the chat function. So make sure you ask your questions in the Q&A section and not using the chat. So during the session, we'll have some questions just straight after the lectures, but then it'll follow by a full Q&A session with all the speakers afterwards. My name is Denny Sakkas, and it's a great honour to be co-moderating this session with Professor Fulvio Gandolfi from Italy. Fulvio.
Thank you, Denny. I'm really very pleased to be hosting this session with you. After a long while, we see each other through the internet, given the situation. Well, you all know Denny Sakkas, but for the few who don't, I will briefly summarize his bio that is also available on the website. He received his PhD at Monash University in Melbourne and has an extensive experience in clinical IVF and reproductive research. This is internationally recognized, having organized conference and workshop on reproduction-related topic. He leads a very productive research team investigating many aspects of basic and translational reproductive biology and has been extensively funded throughout his career. In fact, Denny has more than 200 publications under his belt that span from across the field of fertilization, early embryo development, and male infertility. Thanks to his experience, he serves on the editorial board of many leading journals. Dr. Sakkas is now the Chief Scientific Officer at Boston IVF and consults to a number of clinics and startup companies in the area of IVF. He is an adjunct associate professor at Yale University School of Medicine. Denny.
Thank you, Fulvio. Thanks a lot for that introduction. Well, Fulvio Gandolfi is a professor of veterinary anatomy and embryology at the University of Milan in Italy. I came to know him in the 80s because he developed the first embryo co-culture system with oviduct cells that enabled a lot of use in vitro fertilization in a lot of domestic species. He was a postdoc in Professor Bob Moore's lab at Cambridge. And if you don't know who Professor Bob Moore is, you should know, because his lab pioneered many of the techniques, even though it was an animal lab, but many of the techniques that we use in human IVF today, and in particular stimulation protocols that are even used today. Sadly, Bob Moore passed away last week, so it's timely we can honour him with this initiative and session today. So it's great that we can also think of some of the pioneers in this field in both animal and in human IVF. Fulvio, after working with Bob Moore, studied the molecular interactions between the oviduct and the embryos and characterized a lot of the oviduct-specific proteins that we know now bind to the embryo. His research moved more towards molecular mechanisms of oocyte competence, and he described the polyadenylation of maternal messenger RNA and distribution of mitochondria and actually the metabolic competence through manipulation of cyclic AMP. And you can see a lot of these publications and get more information on the website for his bio. More importantly, he's also been president of the International Embryology Transfer Society. He's current president of ICAR and is currently the editor-in-chief of Theriogenology. And Fulvio will actually give us a short introduction to what Theriogenology is about.
Thank you, Denny, and thanks to the organizer for giving me the opportunity to introduce Theriogenology. First thing first, what does Theriogenology actually means? I learned that not everybody is familiar with this unusual word. The term is the fusion of two Greek words. So one is therion and the other is genesis, which means animal and birth. Therefore, Theriogenology means animal reproduction. This strange word is commonly used by veterinarians, especially in North America, which is where our journal was founded. Theriogenology, in fact, was born in California in 1974, so we are 47 now. Here you see the original cover of what was a no-frill journal dedicated to practitioners interested in the reproduction of domestic animals. However, you can notice that it had an international editorial board from the very beginning. Setting the scene for what it is now, we changed the cover on the 30th birthday, and it now shows the first in vitro mature and in vitro fertilized bovine oocyte. The picture was taken by John Parrish in 1984 and was published in Therio in 1986. We also expanded our interest to wild species, poultry, and fish, but we limit our scope to research somehow linked to veterinary and animal science. Despite this, we have experienced a constant growth of submissions that last year peaked at the record level of nearly 4 papers each day. Unfortunately, more than 60% of them do not make through the cut, but this still leaves us with more than 500 manuscripts published last year. We are a truly open community with submission coming and being accepted from all over the world. Down below, you see the list of 10 biggest contributing countries, and this represents a fairly accurate picture of the world research activity in the field. So spanning from China to Japan, going through Spain, Poland, Italy, Republic of Korea, Iran, and so on. Our readership is also global, and we are proud to provide free access for more than 9,000 institutions in low-income countries. We reward our reviewers with free access to the Science Direct and Scopus services. And probably more important, I think, we support them with free online resources created by experienced editors and scientists for improving their professional skills. The other reason why I'm here tonight, I think, is because both my journal and myself are long-standing convinced advocates on the use of the importance to include large animals into translational research, which is also which is obviously the underlying theme of today's meeting. We all agree that mouse is a beautiful animal and enable impressive research. At the same time, it has some relevant limitation when moving from basic to preclinical studies. Here is an extensive list of topics that can benefit from the use of domestic animal models. I now enlarge just a few of them that may be more relevant to you, like epigenetics and environment, including the effect of photoperiod, global warming, seasonality, and elevation, and the modification of our gene function. And of course, reproduction, including gametogenesis, gonadal function, infertility, and aging. And you will soon have an excellent example shortly from our third speaker. Apart from some unexpected links between Nobel laureates and the use of farm animal models, my personal view on why this species should be considered when planning biomedical research comes down to the concept of better physiological relevance. This can be further specified talking about anatomy and size, metabolism, metabolic rate, lifespan and pregnancy length, heterogeneous genetic background, and certainly many of you can add other items to the list. Along the years, my colleague and I generated some data in this area, both within and outside reproduction. The common pattern has been that the molecules involved in the specific physiological event under study were in most cases the same as in the mouse, but they had different role or slightly different angle. This invariably provided a new perspective and a better understanding of the whole process that we happily share with the rest of the scientific community. But now enough with this general consideration, and let's enjoy the interesting data that will be presented by our excellent speakers that obviously all work on domestic animals. Thank you.
And here we are. So the first speaker, as you know, is Dr. Pierre Comizzoli, and it's a real pleasure for me to introduce you. Pierre is a long-standing friend, a regular contributor to Theriogenology. an appreciated member of its editorial board. Pierre started his career as a research veterinarian in South America and Africa. And after completing his PhD, he joined the Smithsonian Conservation Biology Institute in Washington, D.C. in 2002, where he developed several cryobanking projects on gametes and gonadal tissue from rare and endangered species, and coordinated reproductive monitoring and artificial insemination in rare species like giant pandas and clothed leopards. Since 2007, he is leading a Smithsonian-wide initiative to improve the management and use of the institution biomaterial collection. The title of his talk is Reproductive Aging, Lesson from the Wild. Pierre, the floor is yours.
All right, thank you very much, Fulvio, for the introduction, and thank you for the organizers to give me this opportunity to come for the second time, actually. And let's get started, because first of all, I just want to say that I don't have any conflict of interest to disclose. As Fulvio just explained, this is true that, well, there is a lot of interest in animal models, and the most common one in terms of laboratory research is really the mouse, and we cannot really dismiss really the huge contribution of that animal model. And when we are talking about reproductive aging, first of all, in females or in women, we are talking about a series of different really intrinsic impact of aging of the organism on the level of steroidal hormones that are circulating in the body. Of course, impact on the oocyte number and oocyte quality, which is impacting the conception rates, and then of course the embryo development in patients or even in animals. Then of course problems with embryo implantation, fetal developments, birth defects, and then after that later on, even if all patients or all animals they can make it to the birth, they have issues with lactations, maternal care, and then after that, of course, the offspring, the fertility of the offspring born from old parents also may be impacted. So what's interesting also that, of course, it's affecting males too, but in males, it's a little bit different, because we know that there is a continuous spermatogenesis most of the time. And we have seen a lot of studies regarding the increase in terms of sperm DNA fragmentation with age, either in animal models, lab models, but also in human. And also what's important is to emphasize the impact of environment on both sexes in terms of premature aging, because stress, social interactions, or even pollutions or toxics in the environment may really increase, accelerate really the aging in terms of reproduction. So just a few examples, talking about animal models, not only, of course, large models like Fulvio mentioned, and also some primate models that are usually very common in lab research, but you can see that like in women, you have a depletion in ovarian reserve. So this is something that has been kind of pretty common. But when it comes to really animal model, that's true that we don't want only to focus on laboratory models like the mouse. We also want to think of large animal species. But in terms of models, since a model is just an approximation in order to better understand other species, and including, of course, humans, the more species diversity we explore, and the better I think we can understand really complex issues. We're never too many species is never enough anyway in terms of models. So that's why I think in my area, in terms of studying, wild animal reproduction, where we have really the possibility to observe and to study and to understand a lot of different species, this is fantastic, because, well, we generate a huge amount of data sets related to the physiology of reproduction of wild animals. And then we can make sure that some of those data are translatable to humans. And also another interest in terms of compared to very genetically controlled laboratory animal models is that when we are dealing with wild animal populations, it's a little bit like in human populations. You have a patient-to-patient variation. You have this heterozygosity that is also very important. You don't have a very standard species. You have a lot of variations, and that's very interesting also to study. So of course, besides the fact that wild animals can be very interesting models, of course, it's important to remember that we share the same goals. When we are on this planet all together, first of all, we want to have babies, not only cute babies, but we also want to have healthy babies. And importantly also is that we share the same environment. So it's very important to understand, especially for wild animal populations, what's really happening in terms of the reproductive physiology, and including for aging, because that may also inform really what may happen also to human populations. So going back to more like conservation biology and my own discipline, reproductive science is a very vast area, and we have discovered a huge diversity. And as you can imagine, a giant panda doesn't have the same reproduction as a lion. So we are constantly doing a lot of basic studies, and we compare really the data between species. And with that, with this very basic information, we are able to make very important conservation actions, either in the wild or in captive breeding centers or in the zoos, really to improve the natural mating, but also, of course, to maintain genetic diversity, which is very important for the sustainability of wild population. And then when we are more confident about the biology of reproduction of a given species, then we can think of assisted reproductive techniques like artificial insemination, in vitro fertilization, and embryo transfer. But also, importantly enough, is what we call genome resource banking, but this is related to cryopreservation, where we have the possibility to preserve the fertility of the animals for the future. And in that case, of course, it's like, you know, in human, we can overcome mating difficulty, we can preserve fertility, as I said, and of course, we can better manage the genetic diversity in populations. So let's go back to really reproductive aging, because, well, of course, reproductive aging in wild animals, you have a lot of different scenarios, not only in mammals, but also in non-mammalian species. Well, first of all, you have elephants. Elephants is a very interesting model because they have a lifespan that's almost similar to human. And in terms of reproductive life, it's also very similar because they have a puberty, you know, around the age of 12, 13, 15. And then in females, they have a menopause, really a reproductive aging around the age of 50, 55. And they do have, like in human, a post-reproductive life, which is really important and which is not that common actually in mammalian species. On the other side of the spectrum, you have really this extreme example, what's called the seminal parity. You probably know already that in salmons, that because salmons, they have one breeding season, they have a breeding season, and after that they die. Well, actually in mammals, you have the same. You have this small marsupial mouse from Australia, and males really have the opportunity to have only one reproductive season. And after that reproductive season, it's really the death, you know, that's waiting for them because they are so stressed, they spend so much energy that that's it. It's accelerated aging, but in that case, it's not reproductive aging, it's really aging because of reproduction. This is fantastic. And then, of course, you have a lot of species like reptiles, and that's a picture of a freshwater crocodile from northern Australia. And those are very impressive because not only they keep growing all their lives, but they have apparently no decline in fertility. They don't age. So those are very, very also interesting models. And I was talking about, you know, captive work of animals in the wild, but also the fact that really in zoos and conservation breeding centers, we have this opportunity to observe animals in a much more controlled conditions. And we have, of course, the possibility to take care of them much more longer than they would live also in the wild. So, of course, lifespan, life expectancy is longer for most of the animals in zoos. And so, of course, at some point, you know, they start to age and start to show some symptoms that are not possible to observe in the wild. And this is very interesting for us because we can also generate more data about reproductive aging. So another example, so here, this is a busy table, but for mammalian species, so I just prepared, you know, show you just from rodents to primates. And here on the first column, you see the impact of aging on some kind of the traditional metrics for that are observed in humans. and including, you know, the post-reproductive lifespan. But what's really important to see, first of all, is really the gaps, the research gaps, because there are so many things that we don't know and that we still have to discover. But then after that, what's really interesting is that even within, you know, artiodactyls are really ungulate species like, you know, the cattle, gazelles and antelopes and deer species. And what's really, really interesting is that even within a specific family of animals, they don't necessarily have all the same traits in terms of reproduction and including for reproductive aging. So for example, and the same also for carnivores and primates. And you have in some primates in the wild, a phenomenon that's pretty unique also is the fact that, for example, in wild baboons or in wild chimpanzees, males tend to age in terms of reproduction faster, earlier than females, because females apparently get protected also by the fact that they have multiple successive also pregnancy. Same things also in some carnivores like the red wolf. And in ungulates, as I said, you know, red deer or elk also, some males also tend to age way faster than the females. So this is pretty interesting. And I was talking about the fact that, you know, for elephants, they are known for post-reproductive lifespan, but it's also seen in other mammalian species. Non-mammalian species, also very important, well, laboratory species, you have, you know, the worms and like Caenorhabditis elegans or the fruit fly, the Drosophila, but there are also in nature really other observations. And same, again, when you look at different, I mean, this is not an exhaustive list of non-mammalian species, but when you look at those different non-mammalian species, you can see again that, well, there is a lot of things that we still don't know. But what's really important and really interesting is to see that actually there is a lot of species that do not have really a decline in fertility in non-mammalian species. And this is, again, providing a lot of interesting examples. And of course, for mammalian species and non-mammalian species, of course, we're trying to understand really how to categorize really reproductive aging in each category or in each family of animals. But of course, whenever we are running into exceptions, really, this is very inspiring because it's telling us something that really we want to understand much better. And talking about that, I would like to talk about, you know, the cheetahs, because cheetahs, well, they have a lifespan in the wild that's pretty short. It's around 7 to 8 years old. Of course, in captivity, they live longer. But what we observed in females is that, well, cheetahs in captivity, they keep having, you know, ovarian cycles, you know, most of all their lives. Even when they reach the age of 14, they keep cycling, and which is kind of interesting. There is apparently no decline. However, what we noticed is that when we try to breed them after the age of 8 or 9, or even if we try to do artificial inseminations on those old females, well, there is no way to get them pregnant. So we thought, well, okay, it's maybe like in human then, because maybe the oocyte quality is decreasing and we have a problem of conceptions or poor embryo development. So we did this study where we compared different groups, age groups of cheetahs, and we collected the oocytes from the cheetahs from different age groups. We fertilized those oocytes in vitro. So those are pictures. Of course, we collect oocytes through laparoscopy. So this is the ovary, the type of course, we use the hormonal stimulation to collect in vivo matured oocytes at the metaphase 2 stage. We fertilize those oocytes from different age groups with different frozen semen. And what was very interesting is that regardless of the age group, we were always obtaining the same embryo formation. It's not fantastic. It's not as good as in other domestic species, but the percentage of morula formation and the percentage of blastocyst formation was not declining with age in old cheetahs. We looked also at different, you know, metrics like, you know, the microtubule spindle in the metaphase 2 oocytes. We looked at mitochondrial functions, and there was absolutely no difference. So we were trying to keep understanding, and we were also, as we were doing our oocyte collection in the different age groups, we also looked at by ultrasound at the thickness of the uterine horn. And what we discovered is that when cheetah females are getting older, after the age of 9, 10, they start to develop this they have a thickness of the endometrium. It's called cystic endometrial hyperplasia. And for us, this is the explanation why they are not able to conceive at an older age. It has nothing to do with the oocyte quality. However, they start to develop these kind of conditions in the uterus that are probably preventing, you know, the embryo implantation. Another example, so let's switch to a non-mammalian species. This is a study that was completed with my colleagues from in collaboration with the colleagues of the Migratory Bird Center. So this is a small warbler that spends the summer in Washington, D.C. area, or in the northeast of the United States, and it's a migratory species, and it spends, it's very clever because it spends the winter in the Caribbean. So during the breeding season, when they are with us, we had the possibility to collect sperm. So the sperm of that warbler is kind of interesting. The head is like a corkscrew. It has a very, very long meat piece, and then after that, a short tail. And when you see them swimming, they do not really undulate like mammalian sperm. They go in a really straight pattern. But what was really interesting to see is that when males are aging, older male, they are producing sperm that are way longer, as you can see on that graph, than younger male. And we think that the reason is because the older they get, the more chances also they have to breed, because one female is usually mating with different males. So the males that have longer sperm have more chances also to be the fathers of the future, you know, chicks. So it's very interesting because this aging process there is no declining of spermatogenesis. There is just a change in the spermatogenesis. So we are trying to understand what are the mechanisms, what really triggers this elongation of the sperm. And of course, we are trying to extract really the lessons from this kind of a very interesting case. Another case, going back to mammals, and again, in terms of reproductive aging in males also, it's pretty interesting because in that case, this is a pretty drastic and quick really aging in males in the black-footed ferret. It's a small carnivore from North America that's been saved and now reintroduced in the wild. But what's very interesting, well, usually they have also a very short lifespan in the wild. But in captivity, what we've been able to observe is that after the age of 5, males really have a huge decrease in terms of levels of testosterone, of course, smaller testes, poor sperm production, and also they start to produce sperm that have more and more abnormalities. So this is also a very interesting model to us because it goes a little bit at the opposite of other species where we think, wow, spermatogenesis is continuous, you know, regardless of the age of the male or the man. And so we're trying also to understand really what's really triggering this kind of decrease, not only in the spermatogenesis, but of course, as I said, you know, levels of testosterone and decrease of libido. So again, a very intriguing example that can also take us really to interesting, you know, information for human. So when we are talking again about research areas, you know, trying to be inspired by wild animal species and what can be relevant also to reproductive aging in human, I think we need to keep, you know, looking at, you know, those differences between species that have a very rapid or a completely absent decline in fertility. And as I mentioned, you know, the species that are really pertaining to those different two categories. Of course, I mean, this cheetah model, this is not probably the only animal doing that, but we want to explore so why we have a sustained oocyte quality in older animals, in older females. This is very intriguing because, again, this is very different from what we observe in human. And then, as I said, I mean, what's really interesting with the conservation breeding centers in zoos is that we know exactly, I mean, we record everything about our animals. We know exactly the composition of the populations that we have. We know who's the father or who and what's happening to the offsprings most of the time. But we have a huge database also that would be very interesting to look at because I was talking about, you know, the impact of aging in parents and the impact that it may have on the future fertility of the offspring. And we have the possibility to dig into this kind of a database, really to look at that, but across a lot of different species. And last but not least, of course, the impact of environment on reproductive aging. And in that case, well, we cannot really study all the wild species, you know, in their natural environment, but we can really select some what we could call the sentinel species that could inform us really again about what could be also observed in human. And then, of course, we always, it's not only understanding and accumulating knowledge, it's also trying to mitigate and to find new treatments. So, of course, in animals, there are hormonal treatments, for example, that are already available for aging stallions, but we are trying also to figure out the same for wild species. We are trying also to see if there are ways to supplement, you know, the in vitro culture medium during in vitro fertilization or embryo development. And of course, we are very interested in trying to use animal models to increase really this ovarian reserve during maybe fetal development in most of mammalian species, to try to increase this ovarian reserve and to make sure that females have a longer reproductive life. But of course, you know, I'm talking about here, you know, wild animal models for human, but we cannot really ignore the fact that, of course, the huge amount of information coming from human IVF and human reproductive medicine is also very beneficial to us. So, of course, I've seen some recent papers about the rejuvenation, you know, of ovarian tissues with trying to trigger really the biogenesis of mitochondria. And I think that the next session will be on mitochondria next week. So this is very interesting, and we are very intrigued and interested in using those kind of tools. So in order to conclude, I would like to say that, of course, we value really comparative aging studies, reproductive aging studies, for, of course, for the sake of conservation biology, because we want to maintain our populations in the wild or in captivity. But as I said, there is a huge really advantage and value for human fertility and vice versa. What we really need to do more in wild species is because what we observe and what we try to understand right now, it's more related to senescence, really the general aging of the animals. But there is not enough study on really the germ cell development mechanisms, and we need to do more effort in that area. Of course, male aging needs to be much more studied, and it's not only, you know, in wild animal species, but also in domestic species or laboratory models. We need to do much more research on that, because I think this is still lacking. And of course, as I said before, really trying to select some sentinel species and to understand, you know, what's the impact of the environment really on their reproductive aging. And then, of course, we try to use this knowledge again to provide, you know, new directions and new areas to extend the reproductive lifespan, not only for wild species, but for human reproductive medicine. So I would like to thank you for your attention. This is, by the way, this is a very interesting panda cub that was born at the National Zoo last year from a very old female. We thought that she was done really with reproduction, but we got a good surprise, because after artificial insemination, she gave us this little bundle of joy. And that's kind of again informing us that, you know, well, we keep learning, you know, every day about reproductive aging in our species. I would like to thank my colleague, Dr. Miriam Ottinger from University of Houston, who helped me also to put together those data. And if you want to contact me, do not hesitate to contact me for questions or ideas or suggestions at this email address. And thank you so much, and I would be happy to answer questions later on.
Thank you, Pierre. That was already a fantastic introduction to how we can use information from a lot of the different species to compare to animals. So we'll just ask you one quick question, and I think this is sort of relevant to the translational research from domestic species. Are there species that don't experience reproductive aging, but that also have very long lifespans? Is there anything unique about these species?
Well, yes. I mean, the truth is that we are more intrigued about, the fact that those species like reptiles or the Greenland shark also that are, living for several hundreds of years in the cold water. It's very intriguing to understand what really is the basis of this possibility to have such a long lifespan. But in the meantime, of course, we try to observe and to understand what's going on with their reproductive fertility, their reproductive functions. But yes, there are many examples to take from the wild.
Thank you, Pierre, and we'll come back with some other. You have many questions that are coming through, so we'll come back with some questions later on. So our next speaker is Dr. Virginia Pensabene. And Virginia is a university academic fellow at the University of Leeds, where she moved as a European Marie Curie fellow. She's sponsored by several initiatives and also is working on developing innovative microfluidics systems and nanotools for IVF. So her translational research will be presented today. She was a postdoctoral associate at the Institute for the Italian Institute of Technology, as she's Italian. And you can see more details about her biography online. She's published many scientific articles, up to more than 50 scientific articles. She's also co-founded and is CEO of a highly successful high-tech medical startup that's filed patents in this field, and you will see today that she will present quite a bit of work on translational research from animal and hopefully things that we will be able to use in the future, even in human IVF. So the title of her talk today is Redefining Microfluidics, and it's a pleasure to introduce you, Virginia. So thank you for your talk.
Thank you very much for the kind introduction. Just checking, can you see my first slide?
Yes.
Yes. So yeah. So as you said, I come from the University of Leeds, and my background is in electronics and electrical engineering. So I'm new to this community somehow, even if I've been working on these technology that I will show you in the last 5 years, and I've been working with reproductive groups, reproductive biologists in the last 10 years. So it's so great to be here tonight. So I have nothing to disclose, no conflict of interest. This that you see here is all work related to embryo culture and the possibility to use microfluidics to improve the step of embryo culture, so which is of course influencing IVF success rate. We started this project looking at mainly mice and how to improve these steps for transgenic or genetically altered mice breeding, but we are now starting working with bigger, large animals, so bovine model, looking to applying these to human IVF. And of course, I'm keen to see if there is any interest or changes to this technology that we can apply to use it for other species. So that's quite good to be here. First of all, I want to just stress one more time how important are animals for our research, and specifically for genetically altered animal with mice. Here you see the numbers just for 2018, just for United Kingdom. So this is quite huge if you think about scaling this up to global research. So we definitely need to work properly with these, and to considering that there has been a lot of improvement in terms of techniques to genetically modify mice. It's important to support this technique with the right methods and a better method, easier method to culture embryos from zygote to blastocyst. So the other point that is quite important in genetic altered animal breeding facilities is also the technique that is used for embryo transfer. So the traditional one is the surgical embryo transfer for mice, which is of course quite invasive, and with transgenic mice, micromanipulated embryos, the implantation rate as well as the IVF success rate can be as low as 25%. So there are some issues, and our hypothesis that the culture conditions could be improved. And also, if we look at more refined procedures like non-surgical embryo transfer, this is as well working well with blastocyst, but not consistently used in many labs around the world. So why is this happening? Is it because of the implantation rate of early or not early stage embryos? Is again because of the culture condition. So working on this with Medical Research Council 3 years ago, we started the Crack It Challenge. So a challenge to again have a new tool, a new method to address this problem. So if we look at conventional culture systems, of course, we all use Petri dishes, and the microdrop is the standard, which is good because with improvement of methods for controlling incubators, for measuring pH, for adapting the medium and the medium composition to support different stages of the embryo development, this is quite consistent. There are also slightly different substrates, like the one started with a well-on-well approach that allow to position the embryo and keep it in the same place so that we can use time-lapse microscopy to grade, to follow the development, to select the best ones for transfer. There are however, some things that are not optimized yet. And these are mainly, I said here, has, because especially in training and especially with animal embryo culture, we are not perfect yet. So in clinics, in IVF clinics, we use so many methods to monitor and control all the parameters. In humans, in animals, is not always the same. Microfluidics has been proposed in this field many years ago. And so here there is just a selection of some of the devices that have been published to address the same issue, or also to include the fertilization step all in one chip. The key advantages of these are that there is no oil. So instead of oil to avoid evaporation and to control the fluid, we have instead an additional piece of plastic on top. There is a reduction of volumes because microfluidics means handling liquid below microliters. So all these channels, they have the size of 10 to 100 microns, so basically the same size of a human hair. So if you could put together all of these, there is a lot of potential for technological advancement and to improve control of liquid, to include sensor perfusion systems. But all of these are still at research level. There is not a commercial system that we can use to test and to play with it, to work with it, to see if there is improvement in our embryo culture protocol. Oops. So this is what I developed in my lab. And what you see on the left is the device that sits in a 60 millimeter Petri dish, so one of the common dish that we use for culture. What you see in red are the microfluidic conduits inside the system. And the circle in the center is the chamber where up to 10 embryos can be cultured in groups. We also look at increasing this group number, so it's feasible to grow up to 40 embryos altogether in this tiny chamber. So I won't spend too much time on this, but basically what I want to say is that as engineers and based on this technology, it's quite easy to move from a design to a prototype. So having the right facilities and good expertise, it's feasible in a day to make a mold that we can use to cast every day new prototypes. So you can imagine that having your inputs or collaborating with different groups, we can effectively try different things quite quickly. We use polydimethylsiloxane, which is the most common prototyping material. It's biocompatible in general, and it's transparent and gas permeable. So it's quite nice for microscopy as well as for enabling gas exchange. So the first thing that we did, of course, in the design is to think about how to close and keep the embryos in the right place. So what you see here is the section of the device, and basically the embryos are loaded from the top, going into this channel and reaching this chamber. Once the culture is completed, then they can be aspirated, so retrieved by aspiration from the same channel. And one key point that we consider was to have a very low velocity. So we didn't want to have a huge pressure to load these embryos inside. So this is what happened with the pipette. So in this video, you should see in a couple of seconds, you should start to see embryos flowing from the inlet. Here comes the first one. So what happened is just in one single step of loading, so just with a squirt of the pipette, all the 10 embryos reach this chamber, and you see that they stop immediately and they kind of spread all around in the chamber. The other thing is to avoid stress, because of course we don't want to damage the mechanical integrity of these embryos during handling, during the loading as well as retrieval. So what you see here in blue is the basically zero shear stress all around in the chamber. And the only red areas are the ones where the embryos cannot go, because these are tiny channels smaller than their size. The other thing that we thought was to favor social distancing, since it's it's the right time. So we wanted to have a nice spreading of these embryos to avoid them to cluster, which could be detrimental for their development, and as well as the diffusion of all the compounds in the medium. You see here in up to 8 seconds, we reach a fluidic equilibrium inside the device. This system works in benchtop incubators. So we work usually at 5 CO2 and CO2. We work also with different kind of incubators and gas setting. The key point again is that there is no oil here. So it's just from the initial drop, we just load all of them in one step. We wait for the time, for example, when we reach blastocyst, and we can retrieve them. So it's a very standard thing. We collected a lot of data on these to look at the embryo quality before moving to embryo transfer. And I won't go through all of these, but there is a link at the end to look at the details. The key point for us was to show that this is compatible with traditional method for embryo quality assessment and evaluation. So, for example, looking at blastocyst cleavage outgrow, there is no significant difference compared to the use of a standard 10 microliter drop with 10 embryos inside. So this is key because we want first to start from non-transgenic mice. And first of all, we want to compare our system with something that is working and everyone is using. In terms of metabolism, we have seen a slightly decrease in pyruvate consumption, increase of glucose consumption, which is a good point in terms of implantation rate. We also perform gene expression, so qPCR, to look at gene expression with no significant differences, only some of the genes related to early embryo development were slightly overexpressed, but again, not significant differences. The last point that I want to talk about is an additional experiment that we did, again, to see how compatible this is with many of the experiments that we want to run with embryos. And so we look at analysis of the blastocyst at the end of the culture, as well as analysis of the media. And in particular, we first grow uterine epithelial cells in a standard dish for 24 hours using their own medium, so standard medium for cells, as well as caseome. And then we use these conditioned media to grow these our embryos from one cell up to blastocyst. So from qPCR this time, we have seen some changes in some of the genes that are related to early embryo development. And this is quite nice because we start to combine some of the medium composition with the embryo development and eventually embryo quality. The mass spec analysis is instead a much bigger set of data. So from this one, the key point was that we collected media from the beginning of the culture, the end of the culture, from devices with and without embryos, and all these groups really were really different. So all the compounds, more than 600 compounds that we identified, among these we are able to select the ones that are strictly related to the cell conditioned medium. We can identify traces of plastic, so this is important if we think about translating these into from the prototypes to something in a different plastic, for example, polystyrene. These are all good information to know. And we can also look at different pathways, what the embryos consumed more or less, which pathways were more or less expressed with the different media. And again, all these are available. So I don't want to take too long on these, going through all these hundreds of compounds, but please have a look. I just want to have some final conclusion on these, because talking to many users and many people that are doing everyday embryo culture, we believe that there are some advantages. So there are still some good reason to use and to apply microfluidics to improve these steps. So the group culture is feasible. possible. The other thing is that there is nothing more than this device. So you don't need any other pumps or tubes or any other equipment. It's just replacing the Petri dish in the step of the culture. So it's quite easy to integrate these in a protocol, in a standard protocol. And again, there is no oil and the liquids are much less. There is less washing. So we believe that it could be also good in terms of cost reduction, time reduction during the procedure. As we said, animals are important, but also they are important to translate to human, to innovations in humans. So we are now looking at performing embryo transfer trials, looking at both at wild type that we already started to do, and transgenic mice. We want, as soon as we have we are confident with the surgical, with advantages in surgical embryo transfer, we want to look at non-surgical embryo transfer, which would be a very good target. We started to work with bovine, and as of course, embryo transfer in bovine is all a different story and much more complex to perform. And of course, then after that, possibly have enough data to look at human trial. Just a very last slide, small things that we are doing on top of that, again, to make these more flexible for different application or models. We have made a scaled up device that can accommodate bigger embryos, so large animal like bovine embryos, as well as human, of course. We are working on positioning and being able to track the embryos, so we can inject, we can load single embryos in single places, and then this is being compatible with time-lapse. We are using a very tiny needle, nanoelectrode, that we can functionalize to sense oxygen, pH, and other compounds in the medium surrounding the embryo. And with a collaborator in Edinburgh, we are also looking at manufacturing these with a bioderived, biodegradable plastic, so basically a zero waste device, which we think would be quite innovative. So I just want to thank the sponsors of all this research, as well as my team at the University of Leeds. And I also want, if you want to have more information, and if you would be so kind to give any feedback, just click on this and you will have more information of my research. So that's the end.
Thank you, Virginia. Thank you very much. Very interesting talk. It's nice to see microfluidic back on fashion again after a while. Just a very quick question before we move on and go to the technical side of it all, and many questions will come. Your philosophy, the general philosophy, is that, are you aiming more at having a simplified method in order to have a better standardization, or you think that all this will bring a real improvement of culture conditions, so that you have you can actually improve the viability of the embryos?
Yeah, so it's a great question, because the make it simple, of course, it's the first step. And we have already seen this, for example, training many students every year. They kind of learn much easier to use this technique instead of using a drop under oil. So we think that easier is it's already easier. So it should be also quite good in terms of improving standardization. Once we go through all the steps, so first wild type mice, then transgenic mice, or from mice to bovine, then possibly I will be able to say, you know, it's working for human as well. It's a big step. I'm aware of that. So
okay, thanks a lot. So many more questions afterwards.
Yes.
Thank you, Virginia. And now let me introduce our last speaker of the session. Is Dr. Juliano Coelho da Silveira. a young rising star of animal reproduction biology. After a master's degree in genetics and applied toxicology in Brazil, Juliano moved to Colorado State University in USA, where he received a PhD in biomedical science. His research focus is to understand how ovarian follicle, oviduct, and uterus communicate with each other, and he's studying the role of extracellular vesicles during folliculogenesis and early embryo development. His goals are to understand female reproductive biology, identify new markers, and develop supplements to improve the in vitro production system. Today, his talk is entitled, "Can IVF benefit from extracellular vesicles?" And here is Juliano, please.
All right, thank you, Fulvio. It's Pierre, it's nice to see you guys. It's the first time I'm sharing a section with Virginia. It was nice to be here with you also. I'm really glad to present some of the data that we have been collecting the last years. And so thank you for the invitation. So just for, let me just put a pointer here. And yeah, and just as a full disclosure, I work at the University of São Paulo in Brazil. So we are in the other side of the world, I believe, for most of you guys. And I have no conflict of interest to disclaim. So, all right, let's go. Also, here's my email in case somebody wants to ask additional questions or contact me in the future. Okay, so today my goal is to talk about extracellular vesicles. They are in the news all the time. Even with the COVID, some people are thinking about using them as sprays, nasal sprays, and that's fine if they want to use that. But today I'm talking about them in reproduction. Okay, so I'll talk about the role of EVs in ovarian follicle, how those EVs can work or help in vitro embryo production, and the use of extracellular vesicles for diagnostics. I don't want to take too much of your time, so I'll do a brief introduction of the topics. So this is just a picture of or a scheme from a review paper that we published, I believe, 2 years ago, where we summarized all the papers by that time they use or study EVs in reproductive systems. Okay, most of this paper is focused in the female area, not in the male side. Okay, so just to kind of put you up to date, so there is lots of work about extracellular vesicles present in follicular fluid and how they are present there, facilitating communication between granulosa cells, cumulus cells. Also, as just mentioned, and I just remember that during Pierre's talk, when I was in my PhD, we used old horses as a model for reproductive aging, and we sampled the extracellular vesicles from follicular fluid. And back then, we published a couple markers for oocyte competence associated with reproductive aging. So that's one way the EVs can help maybe IVF. So helping to screening the good or the bad oocytes. Also, there is some recent publications about the role of extracellular vesicles in the oviduct, and so how they can be secreted by the oviduct cells, also how they can interact with sperm cells or with the embryos, okay? And also some recent work from extracellular vesicles secreted within the endometrium or the uterus, okay? And in this case, some of the work is also showing how the conceptus can communicate with the endometrium and tell the mother that the conceptus is there. It's important for maternal recognition, but also how the endometrium can exchange messages with the conceptus and in that way help the conceptus to develop. All right. Okay, So next, what are those extracellular vesicles? So this is a picture from an old paper from 2013, but most of the concepts are still the same from there. So we can divide the extracellular vesicles in different names, different nomenclatures, but mainly the most studied extracellular vesicles are microvesicles and exosomes. So microvesicles are larger vesicles. Usually they are secreted by the budding of the cell membrane. And in that way, they have proteins and also cell intracellular components from the cell inside of these microvesicles. The exosomes or small extracellular vesicles, usually they are originated in the endosomal pathway. So they are generated inside of the cell. They go close to the endoplasmic reticulum, and they can exchange or get proteins and also mRNA molecules and other biomolecules, which are loaded in these vesicles, and then they are secreted from the cells. The secretion, it's a little bit more controlled than the secretion of microvesicles, okay? So once these vesicles, they are in the extracellular space, in the interstitium, they can migrate through this extracellular space or the interstitium or the fluid, and until they reach a target cell. And in this target cell, they can go inside of these target cells using receptor-mediated endocytosis, also fusion between membranes. So in that way, these extracellular vesicles, they can mediate intercellular communication, okay? So these EVs, they have been secreted in serum, milk, semen, follicular fluid. Also, these extracellular vesicles, as I mentioned, they have proteins in their cell membrane. So they have a double layer cell membrane. They have proteins inside of the extracellular vesicles, and also microRNAs and mRNAs. So they can exchange these bioactive molecules. Most of the papers and the tools that we have to differentiate between these vesicles are based on their size, morphology, and contents. So it's really important to always, once you are working with extracellular vesicles, to show what type of vesicles you are working. So based on the proteins, on the size and images, whatever tools you have, it's important to have them in place. So, but let's go to the important topic. So what are our working questions, or what we are following, trying to discover? So as you know, the intercellular communication within different reproductive tissues, it's embedded in biological fluids. So if we think about sperm maturation, inside of the seminiferous tubulus, we have fluids that are produced in there. Also, when the sperm goes through the epididymis, there is also fluid that's secreted by the epididymis or accessory glands, which are important for spermatozoa function. So we have some students here in the lab, a postdoc, which is working in the role of the extracellular vesicles during a sperm maturation, sperm quality, and all of the area. Also, when we think about follicle development, we have the oocyte growing or developing inside of a follicle, which is growing and secreting signaling molecules inside of the follicular fluid. So the oocyte, the COC, it's embedded in a follicular fluid. And upon ovulation, we have the fertilization taking place in the oviduct, mostly inside of the ampulla. And inside of the oviduct and the uterine, we have uterine fluid and oviductal fluid, which are all also around the gametes and also the embryo. And as you can imagine, all of this happening inside of the person or the animal, those messages or those fluids, they are all dynamic. So they can change as the person change. And when we go to IVF, what we have, it's static conditions. So the maturation, the in vitro fertilization, the embryo development, they are all static. So based on that, we formulate two questions. So what are the roles of extracellular vesicles from ovarian follicular fluid? And also, how can the in vitro production of embryos benefit from it? So to answer this question, first, we want to find these extracellular vesicles in follicular fluid, and this is just a typical protocol for isolation that we use in the lab. So we'll be here, and you can, in case you want to perform in your lab, you can do so. At the end, we are going to have small extracellular vesicles. Here we just compare the concentration of extracellular vesicles in small follicles, and this is bovine ovaries, and pre-ovulatory follicles. And what we noticed, and other people also found, that as the follicle grows and is ready to ovulate, the presence of extracellular vesicles within the follicular fluid are a lot less in a pre-ovulatory follicle than in the immature or small follicles. That tells us that communication is really active here and not as active right here in the pre-ovulatory follicles. Also, we performed some Western blot analysis, and let me just minimize here this window so I can, oh no, okay. So here we can see that we have several proteins which are, let me just change this, okay. Sorry about that. And here's just a couple proteins that showing that when we isolate these extracellular vesicles, we have enrichment for ALEX, which is a marker of the endosomal origin of these vesicles. Also, we have PABP, which is a binder of, it's a protein that can stabilize mRNA molecules, which is important for molecule movement inside of cells, and as we can see here, also inside of extracellular vesicles. And here we have a cytochrome C, which is a mitochondrial marker, and we're just showing that when we isolate extracellular vesicles, these small ones, we don't have cytochrome C, which tells us that we don't have a cell contamination. And here we have an image of these extracellular vesicles. This is an electron microscopy imaging, and we can see these extracellular vesicles with a cup shape as they are supposed to be. Okay. So then next we want to make sure that these extracellular vesicles within the follicular fluid, they were coming from follicular cells. So we use transmission electron microscopy again, just to look at the cytoplasm of granulosa cells and look for the endosomal region or endosomal multivesicular body. And we found the extracellular vesicles inside of multivesicular body of granulosa cells, which means that they can secrete those guys in the follicular fluid. And also, when we look at cumulus cells, we verified the cumulus cells also, they have a multivesicular body loaded with small extracellular vesicles, which can be secreted in the follicular fluid. So the follicular fluid, it has a lot of signaling molecules that are well known, but also the follicular fluid, it has these extracellular vesicles, which can mediate cell communication. So one of the questions that we had is if those extracellular vesicles present in the follicular fluid, if they have a signature. And to test them, Ana Clara, which was a master student in the lab, we isolate extracellular vesicles from two different modified endocrine environment. And here we use bovine ovaries. Those they came as a pair from the slaughterhouse, and we could classify the follicles according to the stage of the corpus luteum as a stage 1 and a stage 3. And as we expected, the stage 1, a low progesterone production, and the stage 3, higher progesterone production. And our question was, okay, so since we know that this environment here has a less progesterone production compared to the stage 3, is there the same within the follicular fluid? And as you can see here, we validated that. If we aspirate the follicles and sample the progesterone inside of this follicular fluid, we have a lower concentration in the follicular fluid from the stage 1 follicles compared to a higher concentration in the follicular fluid from the stage 3 follicles. So we have two environments, low progesterone and high progesterone. So our next question was, okay, let's isolate the EVs from these follicular fluids and look for the microRNAs. And as you can see here, we found a bunch of microRNAs, and interestingly, we found 161 microRNAs, all upregulated in the low progesterone environment. So the follicular fluids that came from stage 1, they have 161 higher or upregulated microRNAs. The bioinformatic, it told us that those microRNAs, they are important, or they are regulating. And microRNAs, usually they work as targeting mRNAs and leading them to degradation or repressing translation. So all these microRNAs, they are involved in progesterone-mediated oocyte maturation, oocyte meiosis, RNA transport, and protein processing in ER. So that means that those microRNAs, they have a role, or they can have a really important role inside of the follicular fluid. So, okay, so what those EVs can do to the oocytes? And we asked the question, would the extracellular vesicles supplementation during maturation affect cumulus oocyte cells? So to answer that, first we want to see if the cumulus cells can uptake the extracellular vesicles. We have EVs in red, the nuclei from the cells in blue, and the actin filaments staining in green. So as you can see here, with 4 hours after supplementation, we have extracellular vesicles localized inside of the cytoplasm of cumulus cells, but also in the TZPs, which are the transzonal projections that communicate cumulus cells with the oocyte. And that was really interesting. With 9 hours, we can see some of the green dots at the end of the TZPs. So we thought that was interesting, and we asked how that happened. And here is a video where you can see all the red EVs that were added to the culture. In green, we have the actin filaments, and those some extracellular vesicles close to the transzonal projection, and they are moving towards the oocyte. Can you still hear me? I just had a message for the unstable connection.
Yes.
Okay, thank you.
Yeah, you're okay.
Okay, thank you. So next, we asked the question, okay, if we add these extracellular vesicles to in vitro maturation, would they change the transcriptome of the cumulus cells? And we performed this experiment in a really short time. We aspirate the COCs from bovine ovaries and add the treatments, supplementation with EVs from low progesterone environment and supplementation with EVs from high progesterone environment. And after 9 hours of in vitro maturation, we collect those cells and perform RNA-seq. As you can see from our control, we have the groups moving in 2 different directions. So that means that the extracellular vesicles supplemented to the cumulus cells, they can change the cumulus cells transcriptome profile. So I will not extend this topic. I have 2 more experiments to show to you guys. So, but indeed, the EVs, they can modulate cell survival, proliferation, and protein synthesis after 9 hours of supplementation. So next, the question was how the IVF can benefit from that. So we asked what type of mRNA the EVs they were carrying. And when we did the RNA-seq and performed bioinformatics, we found lots of mRNAs that were involved in chromatin remodeling and controlling gene expression. Among them, we have DNMTs, EHMT1, and histone deacetylation mRNAs. So we asked the question, okay, so if we supplement those extracellular vesicles from follicular fluid during oocyte maturation and early embryo development, they could possibly help embryo development, blastocyst rates, and change, who knows, epigenetic marks within those embryos. So to test the hypothesis, we picked up one microRNA that was highly expressed and supplement the bovine oocytes before the major genome activation. And after supplementation, we can see that indeed the microRNA that's highly expressed in the extracellular vesicles, upon supplementation, it's highly expressed in the embryos. So that means that even once I have an embryo and I supplement with extracellular vesicles, I can have those EVs probably exchanging material with this embryo. So this is just an image to show the EVs within the blastomeres. Okay, so we asked the question, does that help blastocyst rates? And as you can see here, once we supplement the maturation and embryo development with extracellular vesicles from follicular fluid, we have an increase in blastocyst rates. That's not much, but it's important. But our question is about molecular chains. And the interesting thing, we look for different epigenetic modifiers, but we saw the DNMT3A increased in embryos that were supplemented during maturation and embryo development with the extracellular vesicles from small follicles. So that's interesting. So, but does that do anything? So we ask the next question, and we look for change in global DNA methylation and change in global DNA hydroxymethylation. And as you can see here, this is a huge data set, but in summary, when we supplement the oocytes during maturation and the embryos during development with the EVs from the small follicles, we can see that we change DNA methylation, global DNA methylation in comparison to control. And also we are able to change the global DNA hydroxymethylation in comparison to control, which makes us to think that the supplementation with the EVs can really change DNA methylation and hydroxymethylation. So 2 more projects and I'll be done. So how the IVF can benefit from that? So we create a model here where just changing oxygen tension in the medium, in the incubator, we can induce change in global DNA methylation, lower oxygen tension, decrease the DNA methylation. And when we look at extracellular vesicles in the media on day 3 and day 7, the levels of extracellular vesicles, the number of particles, they are changed within the media. So if we have a low oxygen tension, a higher level on day 3 and a lower level on day 7. Interesting, we have this microRNA, MIR210, which is a marker for hypoxic environment. So it's highly expressed in embryos exposed to low oxygen tension, and also highly expressed in extracellular vesicles secreted by those embryos. So that means that once we have embryo culture and we sample the culture media, we can look for markers of embryo quality. And I think that's really nice and a nice way that IVF can benefit from extracellular vesicles. And the last question, we are jumping from the ovarian follicle and going to the oviduct. And here we created a model, and those are Nelore cows, those are Bos indicus. Most of the cows we have in Brazil look like that. And we single embryo, and some animals, they were cycling, and they were not pregnant. So we slaughtered these animals 4.5 days after induction of ovulation, flush the oviducts and collect small extracellular vesicles and confirm if they were pregnant or not. So just to show you the embryo at the 4.5 days after induction of ovulation, embryo should be at the isthmus where we have lots of ciliated cells. So when we look for microRNAs inside of the oviductal epithelial cells and microRNAs inside of the extracellular vesicles secreted in the flushing or the oviductal fluid, we have a different profile of microRNAs, and you can check all of that in this paper just recently published. So we perform bioinformatics, lots of pathways involved in cell proliferation, inflammation, immune system, and maternal recognition. But we choose a couple genes just to test if a single embryo could modify the environment. And as you can see here, we have the pregnant animals inducing an increased expression in TGF-beta1, TNF-R1, and PTGS2. So that tells us that communication between the embryo and the mother already started at the oviduct, and the embryo can tell the mother to induce change in anti-inflammatory response, contractive substance, and also immune tolerance. So to finish, we are creating a model which we are trying to develop here in the lab, where we believe the IVF can benefit from extracellular vesicle supplementation during oocyte maturation. And we have 2 steps, so would be EVs from small follicles and from large follicles. But also during embryo development, we believe that embryo development could benefit from extracellular vesicles coming from the oviduct. Of course, this is in the bovine and the animal industry, not in the human side. But why not? Follicular fluid could be good also supplementation additive for in vitro embryo culture. So I would like to thank all my collaborators here in Brazil, also in Canada. This is my email in case you want to contact me. Those are the financial institutions that support all this work, and some of our partners that help us donating semen and other supplements for embryo production. With that, I would like to thank you for the opportunity, and I'm open for questions.
Thank you, Juliano. That was fascinating. I'll have one question for you, and then we'll open up the session to more question and answers to the 3 speakers. We'll do that for about 15 minutes, and then we'll close the session. So I have one question, Juliano, that obviously you've shown that there is an effect from the extracellular vesicles, but on the contrary, we do a lot of things in vitro in many of these models where these vesicles are not present. So the role, how would you describe the role of these vesicles? Are they fine-tuning the system, or are they, are they crucial? How do you define their role in some of these systems where they're actually probably not present?
Yeah, so thank you for the question. And so in my point of view, this type of cell communication, it helps to fine-tuning in the system. So in that way, even when you mention that they are not present, it's because we don't have the mother, the female reproductive tract. But once we think, if we think about embryo culture of a group of embryos, we have extracellular vesicles secreted between one embryo and exchanging information with the other ones. So in a type of setting where we have a fluid exchange between one embryo chamber and the other one, we can have these extracellular vesicles helping kind of fine-tuning this system. But I believe they do this fine-tuning and kind of real-time getting the system up to date to work and produce this viable embryo.
Thank you. So let's go back to the first speaker, to Pierre. Fulvio, do you have any questions for Pierre?
Well, I see that there are quite a few questions from the public from Pierre. One of them is, do you know what is the minimal threshold, is any, of species population size to maintain genetic diversity?
Okay. Yes, we have models, mathematical models to estimate that. So based on the current existing composition of rare and endangered populations, we know exactly how much was the target in order the number that we would have to reach in order to maintain more than 90% of genetic diversity for the next 100 years. What I can tell you is that most of the time we are below that target in a lot of species, so we have to work really hard. But yes, we definitely know exactly how many animals we need.
Oh, great. And then there is another one related. Well, it's about our closest primate relatives. Do they age like we do? Are they any different, or are they the best model as we think, or not?
Well, yes, well, but the primates are also very diverse. Of course, they are closely related to human, and they can be very, very useful. But the truth is that, as I explained also, is that some species are showing very traits in terms of reproductive aging that are very different from what we see in humans. And I was referring to aging in males, which is apparently more prevalent in some, and happens earlier in some species, primate species, than in human.
All right, great, thanks a lot. Denny, do you want to ask?
Yeah, Virginia. So the microfluidics is fascinating, and I think it will lend itself to in vitro fertilization, and there are a lot of groups working on this, as you know. I had one question. So one thing that has been some people have been questioning for a long time, and is very pertinent between species. Embryo grouping or clustering, it's favorable in some species, especially in the bovine, where it's really needed. But some other species really don't seem to use it. You seem to have shown a system where distancing helps. Can you just comment on grouping, individual culture, and how you're answering those questions using the microfluidic systems?
Yeah, so it's a quite interesting and long story. So I guess the first studies are from the 70s, looking at the appropriate distancing between embryos. And there is a balance, of course, considering the distancing and also density per volume. So I guess the point of spreading is to avoid excess of too much waste all around the same embryo. But it's true, as Juliano said, that we also want to have embryos close enough so that they can release and fill the others. So in our the first step for us was to keep them far enough to monitor them, to look at the distance, how much they move reciprocally, and also there are as well more new data where they are looking at the appropriate distance, and I guess that fed all the design of well on well and grids, for example, for bovine embryos, so where the control of distances is much more important than mice.
So as a bit of a follow-up to that. The system you showed us was more of a static system, whereas, one of the beauties of microfluidics is that it can be made a dynamic system, and you can change media and maybe use the media to go with the different stages of embryos. So are you also looking at more dynamic culture systems?
Yes, so the static is because, as Fulvio asked before, static it's easier, and for a static system you don't need to have pumps or other tubes going in the incubator, things that can make the protocol complex. So we like to study the static first, and in mice the single medium is quite common. So we are looking now at changing media, as you said, to support different stages of development. It's absolutely easy to change media in a microfluidic system. You just flash new medium through the system. So that's a key. That's a good point as well. Of course, changing embryos maybe not for mice.
Here I was trying to unmute my microphone. Sorry. I read an interesting question for Juliano by Debbie Mongin. Do you think we will have a synthetic or manufactured vesicle to add to our culture medium in the future? Is a pathway that you think we can go towards actually making our own microvesicle instead of going through the complex methods of actually isolating them and standardizing them as well?
Thank you, Debbie and Fulvio, for the question. Yes, I believe that's the future, and there are some groups that are already working into developing these vesicles, and that's the only way we could use them in IVF to make the system secure, if we think about domestic animals, but also if we think about helping the human IVF, there could be something more controlled. Of course, I don't know if there is a legal concerns if I'm using my own body fluid to help my gametes. I don't know if that will be a concern. So if I have to mature my oocytes in vitro, so why not the follicular fluids there? So why I could not use that? But one important question for that we have to answer before the synthetic vesicles, Debbie, is to know what we want to load in these vesicles. And that's what my lab is doing. So we try to find proteins, lipids, microRNAs, mRNAs, what kind of molecules do we want to put in these future synthetic vesicles? And that's what we are pursuing here. Thank you for the question.
Right. Thank you. Then there is another one. I don't know if you have the data for this, but maybe you have a feeling for this. So do the vesicle profile changes during the cycle in the oviduct and the uterus? So have you already have the opportunity to see if the cycle has some effect on the vesicles?
Yeah, so we are working in a project right now where we have a good amount of data on that to show that the cycle can affect the contents of the extracellular vesicles. Not much in numbers, but also, especially in the contents. Also, when we look in the follicular fluid, and that was, I was mentioning in the oviduct, in the uterine fluid. But when I mention, I'm looking at the follicular fluid, as I mentioned before, if I just look at follicular fluid in stage 1 or stage 3 of the estrous cycle, I can see lots of change in the microRNA profile within these vesicles. And to test that, we just supplement that, and we can change the transcriptome profile. And that, for me, it's amazing.
Thank you.
Pierre, I had a question. I loved your slide with the sperm length in the birds, and birds have very interesting evolutionary competition, let's say, in terms of the way they mate. I'm interested, do you have this type of data on males that are in polygamous or sort of mono species where they only have one partner in a lifetime versus those that actually have more competition where the female has multiple partners? So could you comment on some of that data in terms of sperm?
Yes, that's a good point. And actually, the data that I showed about the warbler, that warbler is a monogamous species, but they also, it's a monogamous species, but they have what we call the extra-paternal, pairing. So they go see around, around a little bit, but it's definitely yes, and it's probably in other species that are not monogamous. It's probably even more striking about the difference in terms of sperm size. Yeah.
To continue on the sperm theme, I just wanted to ask actually Virginia, you were working a lot on embryos and that, but microfluidics is like really lends itself to sperm just because of the way they move and that. So are you doing, and do you have any comment on sort of the use of microfluidics in sperm research or treatment in different species?
It's not my expertise, honestly. So I have never worked with sperm myself. So, but I know literature is huge in that field, and in that sense, I have to say it's quite interesting for me to see that there are products already or new technologies that have been translated for sperm, but there is nothing yet for embryos. So it's quite interesting because embryos are bigger, so it should be easier from the engineering point of view to make a microfluidic device that fits more static culture while compared to sperm. So that's all I know, unfortunately.
Can I ask you another question, Virginia? Well, actually relating to one of Murray Gallagher asks. So the question relates how your system behaves in comparison with the oil drops in terms of buffering the medium. So the oil also has the ability to provide some buffering of the changing of the situation of the medium. Do you obtain the same result with your microfluidic system, or is more susceptible to external variations?
Yes, so the PDMS, this plastic that we use for these prototypes, is permeable, so it absorbs small molecules. It absorbs estrogen, it absorbs many things that we ideally want to keep stable and under control in the medium. So PDMS is not the best for these devices, and that's part of the prototyping phase. So we hopefully we will move to something more traditional like polystyrene. Compared to oil and the point of buffering, we compared with mass spec as well, more standard in a more standard experiment, just the device compared to the oil, to the micro drop, and there are differences, not huge, and again we cannot really correlate changes with changes in the embryo development, because as you have seen, they're kind of growing in the same way. So even if there are changes, we don't know if they are effectively important or needed.
Yeah,
I think
well, where you are at it, just a very quick one. So yes. You said that that PDMS absorbs things. Can you just do it as we used to do with oil, just saturate it with medium and then change it all? So let it saturate the plastic like we used to do with oil.
Yes, kind of. So there are steps that I didn't mention, but there are steps to prime the device before loading the embryos. So you first do several washing with water, for example, for a long time at temperature, controlling temperature as well to favor or block this absorption. And then you can do sort of saturate some of these compounds in the PDMS, but it's not, again, PDMS is the way to go to test different things, but it's not the final one. That's probably why many things, that's my opinion, why many things didn't go farther than this prototyping phase.
Thank you.
So just one last question for Juliano, hopefully a quick one, and we'll wrap up. Juliano, there's a lot of interest in the human now of actually finding DNA in the culture media and using it as a marker. In your studies, have you been able to find DNA in vesicles?
So that's an important question, and it's also controversial. You know, if we think about the size of the vesicles, it's kind of it's hard to know if the DNA is within the vesicles. If we think about a nuclei, the nuclei from the cell has 5 microns, and we have all the DNA packed inside of 5 microns, and EVs are 30 nanometers. 100 nanometers. So, but I have not tested, and but maybe the DNA is around the vesicles as a subproduct of apoptosis or something like that. Because if we think about a cell membrane, as the cell membrane and the EVs, they also have sugars and things stacking outside of the cell membrane. So why not we can have DNA wrapped around these sugars domains? So that's a good question.
Thank you. Unfortunately, you know, we still have a lot of questions, and it's been a great session today. And I'd like to thank everybody for taking time to participate in the session and the discussion, and of course, all the participants who asked the questions. Of course, I'd particularly like to thank Pierre, Virginia, and Juliano, and also it was fantastic seeing Fulvio again. And so as a co-host, I'd like to thank Fulvio too. Last but not least, this doesn't just happen out of thin air, and I'd like to thank the organising committee for this initiative again, for the I3 initiative. Jacques Cohen, Peter Nage, Thomas Elliott, Shaista Sadrudan, Giles Palmer, who does an amazing job of herding everybody together, Marianne Saivetes, and today's team who were in the background that you didn't see, actually organising the question and answers from everyone. And there are a lot of questions coming in. Alison Bartolucci, Marianne Saivetes again, and also Shaista Sadrudan. Thank you.
Thank you, Denny. Thank you, everybody. Thank you all to the more than 400 attendees that listened to us. That was a big surprise to me. A very popular session here. Thanks a lot to everybody. And of course, for those who were unable to stay with us live, we remind you, everybody, that session and presentation will be available on the website soon. So, after all, don't forget to rate our lectures as well. Your certificate of attendance will be sent before commencement of our next session. And our next session will be on Tuesday, the 23rd of March, as always, at 3 p.m. Eastern Standard Time. However, be careful, due to clock changing, there may be some 1 hour difference in many regions. So double check your timing. With moderators, Professor Elia Adashi and Dr. Jack Cohen, the next session is entitled The Mitochondriacs. So this, in this session, we will learn about mitochondrial reproduction and how to prevent the transmission of mitochondrial DNA disorders using pre-implantation genetic diagnosis with 2 great speakers, like Professor Bert Schmitz and Professor Justin Singin. So that's all for tonight. Bye-bye, and see you next week.